Hello everyone, this is Kartika from Department of Biotechnology. I am here to tell you all about the lacopron. Lacopron is a set of genes present in the bacteria which codes for enzymes to break the lactose. Before getting into the details, let's see a friend of ours, the E. coli. Our friend E. coli lives in our intestine. Yes, our intestine, and they love glucose. Glucose is a simple sugar, gives energy to E. coli. If we didn't feed our friend glucose, he uses lactose, a disaccharide sugar which is made of glucose and galactose to gain energy. Breaking lactose needs energy. That's why E. coli always choose to eat glucose because it's readily available. In case of no availability of glucose, he had to use lactose. For that, lacopron produces enzymes to break the lactose, but they need to meet some conditions. Let's see them detail. As I mentioned before, lacopron, a set of genes, contains a promoter, operator, 3 structural genes, and 1 regulatory gene. Promoter region is the place where RNA polymerase binds for initiating transcription. Operator has the switch of lacopron which can be turned on or off. Regulatory gene produces a repressor which keeps the operon off. Lacopron has 3 structural genes which produces 3 different enzymes. LacZ codes for beta-galactosidase which breaks the lactose sugar. LacY produces permease enzyme which helps in transport of lactose into the cell. LacA synthesizes trans-acetylase whose function is not completely studied but some suggest that they involve in detoxification. The regulation of lacopron is controlled by the presence or absence of lactose and glucose, and also the level of glucose plays a vital role. In the absence of lactose and in the presence of glucose, the operon remains switched off. That means no transcription occurs as a repressor made by a regulatory gene was bound to the operator, blocking RNA polymerase. As I said before, E. coli love glucose, which is readily available, so they can use them to gain energy. In this state, operon is repressed. Meanwhile, in the presence of lactose and in the absence of glucose, a co-repressor called allolactose binds to the repressor, making it inactive to bind on operator. This switch off the operator. Hence, the transcription occurs and the enzymes are produced to break lactose. In this state, lacopron is induced. A protein called catabolite activator protein, in short CAP, is like a friend of RNA polymerase which helps the polymerase to bind to the promoter and increases transcription rate. If the glucose level is low around the operon, a messenger molecule called cyclic adenosine monophosphate, to say easily, let's call it CAMP, is produced, and it activates the CAP by binding to it. This increases the transcription rate. While in case of high glucose, the CAMP is not produced, hence low transcription rate. The summary of lacopron responses is given here. Take time to look at it. And that's all for lacopron. Thank you for listening.
I am from the Department of BSc Biotechnology student. Now I speak about the topic of prokaryotic translation machinery in the subject of molecular biology. Introduction. Translation is the process of protein synthesis on mRNA template. Definition. Translation can be defined as the process in which mRNA is utilized. Factors involving in translation: ribosomes, amino acids, mRNA, TR. Ribosome, functionally active ribosome, prokaryotic ribosome, 17S ribosome. tRNA, made up of 70 to 90 nucleotides, clever leaf shape. Various process of translation: initiation of translation, elongation of translation, and termination of translocation. Activation of amino acids. Amino acids in the cytoplasm occur in the inactive form and they cannot take part in protein synthesis. Transfer of amino acid. The enzyme bound activated amino acid aminoacyl adenyl. Initiation. Translation begins with the association of 13S ribosomal Elongation of polypeptide chain is brought about by the regular addition of amino acids. Determination, third phase of translation. Protein synthesis is signaled by one or more proteins. Conclusion, translation is the process in which a polypeptide chain obtained of different amino acids.
Hi to all. I'm from Department of BSc Biotechnology student. Now I'm going to speak about replication origin and replication fork on the subject of molecular biology. Now I'm going to part 1 introduction. What is DNA replication? It is process by which make a synthesis of new DNA strand. So the process it should be the start from some site. So this question about where is the DNA synthesis start? The answer is origin of replication. The replication fork is a very active area where DNA replication takes place. Part 2 origin of replication. In prokaryotic the replication origin of replication in single site, whereas in eukaryotic the origin of replication takes place at many. Enzymes involve the origin of replication. DNA helicase, DNA polymerase, RNA polymerase, DNA ligase. DNA helicase catalyze the descriptions of hydrogen bond that hold the two strands of double-stranded DNA together. RNA polymerase accurately and efficiently replicate the genome in order to ensure the maintenance of genetic information. RNA polymerase click here to add content of the text and briefly explain your point of view. DNA ligase click add to content of text and briefly explains your point of view. Part 3 Replication Fork. Replication fork, the open area of DNA where replications or transcription can take place, DNA helicase. And when the DNA double helix to from single strand of DNA, the unbound region of DNA molecules is referred to as replication fork. Formation of replication origin DNA helicase unwound the DNA replication fork formation. Thanks to
Transcription. It is the process of synthesis of one RNA molecule using one of the two DNA strands. It uses two DNA strands as a template by using the enzyme RNA polymerase. Eukaryotic RNA polymerases RNA polymerase 1, 2, and 3. RNA polymerase 1 transcribes and synthesis. 2 and 3 transcribes. It contains 28 as rRNA, 18 as rRNA, 5.8 as rRNA, and 2 contains mRNA, some snRNA, and RNA polymerase 3 contains tRNA, 5 as rRNA, some snRNA. RNA polymerase 2. RNA polymerase 2 is defined as the central to eukaryotic gene expression, and RNA polymerase 2 is a multi-subunit enzyme which has 12 subunits, and RNA polymerase 2 requires an array of other proteins called transcription factors, which is helpful in forming the active transcription complex. Eukaryotic transcription promoters, which are more complex than those found in bacteria. These are sequences which is located in the upstream regions of coding strand. Mutation of this region usually significantly lowers the rate of transcription. Tata box, which is also known as Hogness box, which is similar to the prokaryotic Tata box, but the sequence is slightly different. It is located in between the scale minus 25 to minus 30. Cat box, which is located in between minus 70 to minus 80, which always contains double C, double A, T, the sequence. GC box usually has the sequence of triple G, C, double G, and it is typically found at minus 110. Termination. When the RNA polymerase transcribes the terminator region of the DNA, the polymerase releases the mRNA. The termination sequence is WAT, AAA, followed by GT repeats. Termination. When the RNA polymerase transcribes the terminator region of the DNA, the polymerase releases the mRNA. The termination sequence is WAT, AAA, followed by GT repeats. Elongation. TF2F is a factor which remains associated with RNA polymerase 2 throughout the elongation process. The activity of the RNA polymerase II is enhanced by the proteins called elongation factors. Termination. At the terminator region, the RNA polymerase releases the mRNA. The termination sequence is AA, T, and AAA.
Molecular biology type of RNA present by J. Rojapanu. RNA or ribonucleic acid is a polymerase of nucleotides that is made up of a ribose sugar, a phosphate, and base such as adenine, guanine, cytosine, and uracil. RNA has structure very similar to that of DNA. Three types of RNA. mRNA. mRNA accounts of just 5% of the total RNA in the cell. The mRNA is 5' to 3' direction. Molecules of mRNA are composed of relative short single strands of molecules made of the adenine, cytosine, guanine, and uracil based, held together by a sugar phosphate backbone. Ribosomes read the mRNA and translate the messenger into function proteins in the process called translation. mRNA and hnMRNA, monocitronic mRNA, bicitronic mRNA, polycistronic mRNA, and transfer RNA, tRNA. tRNA is the smallest of the three types of RNA, possessing around 75 to 95 nucleotides. tRNA are as essential components of translation where their main function is the transfer of amino acid during protein synthesis. Therefore, they are called transfer RNAs. tRNAs also act as adapter in the translation of the genetic sequence of mRNA into proteins. Structure of tRNA. rRNA, ribosomal ribonucleic acid. rRNA is a type of non-coding RNA which is the primary component of ribosomes essential to all cells. RNA is a ribosome which carry out protein synthesis in ribosome. Ribosomal RNA is transcribed from ribosomal DNA and then bound to ribosomal proteins to form small and large ribosome subunits. rRNA is a physical and mechanical factor of the ribosome that process transfer RNA and messenger RNA to process and translate the latter into protein. Ribosomal RNA is the predominant from the RNA found in most cells. It makes up about 80% of cellular RNA, despite never being translated into the proteins itself. There are types of RNA: mRNA, tRNA, and rRNA. Thank you.